Hi everyone.
Hi everyone,
Today I'm going
I'm
to
Saranya Dura
explain my
Samantro,
project,
and my
Big
final
Data
project title
Predicting
is I
Genetic
took the data
Variability
study in
via
life
Machine
fall
Learning
semester, and
Algorithm.
for my case study, I built a machine learning algorithm that will predict
My
genetic
final
mutations.
project title is
So
Big
I'm
Data
going to
Case
explain
Study
one by
in
one
Life
slide, and I'm
Sciences.
going to provide
For
some more
my case study,
information.
I
And
built a
I'll
machine
also show
learning
a mini demo
algorithm.
that will predict genetic
And let's
mutations.
see.
Coming to genetic mutations. So, what are genetic mutations? Genetic mutations are something when your amino
Genetic
acid
studies
sequence
and
changes
testing
or
have
something that
transformed
causes tumor. Treatments of many
a
genetic
tumor can
disorders,
have thousands
including
of
cancer,
mutations
once your gene is
a gene
sequenced.
is sequenced.
But
The hard
I mean, a
part is
tumor gene
finding
is
the
sequenced,
normal
it can have
mutations
thousands of genetic mutations
from the tumor
but
causing
currently, the
mutations.
challenge we are
Not
facing
all
is
is
distinguishing the tumor
tumor
causing and the normal
Currently,
mutations.
a mutation can be
Interpreting
classified
mutations
into 9
manually is a
classes,
time-consuming
but
task,
the
which calls for the
time-consuming part
development
is
of a
most of the
machine learning
classification is
algorithm,
done manually,
which will be able to
by
classify
searching
genetic
like a lot of
mutations based on
literature and
clinical
real
information
evidence
or
or
clinical evidence. So a lot of time is being wasted in searching
The data
manually
set,
for each and
the
every
genetic
mutant.
mutation can be
And when your
classified
mutant size is
under
like
9
high in number,
classes,
then
and for this particular
searching becomes
project,
like a very difficult part.
the data set
So
includes
the
both
machine learning
training
algorithm
and test
described
data
in this
files
project
for the
uses the
variations
clinical
and clinical
evidence as a
evidence.
knowledge base.
So the training and
The
test
model
variance
trains using the
provides the
clinical
information
evidence.
about the
And
genetic
what I have did
mutation,
for this was I
whereas
divided
the
the
test
data
data
set
set
into
includes the
training
clinical
data set
evidence provides the
and the test
information
data set or the clinical
so the
evidence
data
that are
set
used to
includes both
classify
training and test
the
data
genetic
files for
mutations.
both the
So
variations and
the
clinical
primary
evidence.
key or the
So the
common
training
field
and
that
test
are used
variance
to
provides the
link
information about the
the
genetic
text or
mutation
the clinical evidence and the variation file
the
would
text
be the
the
ID
text
field,
file provides
which
the
is
clinical
serves as the
evidence
primary
that are
column.
used as a knowledge base
And
to
I have
classify
provided a
the
link to the
genetic
dataset
mutations.
if you want to download the
So
data and run the code. So this machine learning algorithm, in short,
So
takes
the
the clinical
technology.
evidence as a
So the
knowledge
machine
base
learning
and
models
it
were
classifies
used
the
using
mutants
both
into
R
one of
and
the nine
Python.
classes.
So
for R, I have
two
used
models
the
for this
XGBoost
package.
package,
One is the
and
XGBoost
which is
model
used
from
which was
R,
used to
and one
develop
is
the
the
model.
Keras model
And
from
for Python, I have used the
So
Keras package
please
or the
take a
Keras
look at my
module
project report
using
if you
which
want
I
to know more
developed the
about
machine learning
this model.
algorithm and the training
So some
accuracy
of the
and
techniques
predictions
that I have
from both the models
thank
included
you.
or implemented in this project would be so importing data files and natural language toolkit to remove stop words and like sparse items from the data set, and developing the machine learning algorithm using different models. And so XGBoost would be R. So and I also included a word cloud to identify the most frequently or commonly used words in the clinical evidence. And then there is also a graph using the matplotlib library to visualize loss and accuracy. So going detail into the data format. So the data files are both comma separated. So they are like, so there are four data files and two are comma separated files and two are like, double pipe limited, delimited files. So to explain more about like the data and variables, so the ID field would be the ID of each and every row, which is the unique or the primary column, and the gene is the gene where the genetic mutation is located, and the variation column explains the amino acid change for a particular mutation. So each and every human being has a particular amino acid sequence, but if one word or one alphabet changes in the sequence, that's what causes a mutation. And class would be anywhere between the numerical value for the class should be 1 to 9, and under which the genetic mutation has been classified. And the text is nothing but the clinical evidence that is used to classify the genetic mutation. Okay. So for this particular demo, yeah, if you want to run this code, mainly you would have to have R and RStudio installed on your laptop. And so there are like some packages mentioned. So you would need ggplot, ggthemes, and the main package, other packages you would need would be xgboost, string R, wordcloud, ggplot, tidyr, and readr. So these are the main packages that you would be needing if you need to run this code. So let me see if I can quickly open my R code. Okay, so as you see in this code, so first I set up my working directory space, and then I load all the packages that I need for running my script. So the first would be loading the data sets. and I upload the train text, which is my training data set, and then I upload my test data set. So once I have my data files upload, I have this, I'll run this quickly. And then I proceed with the language processing methods where there is some data cleanup included. So in this particular text of code, so what I aim to do is I have to remove the trailing space, white space, and then I have to transform all the lowercase. I have to transform all the words into lower cases and then remove the punctuations and remove stop words, which are these stop words that are identified in the clinical evidence, and then I remove all the numbers. So, and then I create a document term matrix. And then once after I create the document term matrix, then I convert that. I then calculated the word frequency to find out the most frequent words. So the most frequent words would be, as you see in this graph, the most frequent words in the clinical evidence were cell and then mutant, which is not that surprising. Okay. So once after I created the bar chart, I moved to go and create the word cloud to see if the bar chart results match the word cloud results. And again, you can see the most frequent words in the clinical evidence were like some of the most clinical. Cell, mutant, tumor, gene, domain, patient, cancer, and active were some of the most frequently used words. And so going to the most important part, model. Yep. Okay. So my data set has 3321 observations for which I divide the data set into test data and train data. So for my train data, I take the observations from 1 to 3000, and for my test data, I take the observation from 3001 to 3321, which would be like 321 observations for my test and 3000 for my train. So I run the XGB model. So initially, the data set is framed into a dense matrix or a D matrix to identify the complex relationship between the features and the labels. So the model was trained using the trained data set, and the class of the test data set was predicted by applying the XGBoost model. And the model was run thrice to predict the value of the class variable, and it found the mode of all the three results obtained, which decided the final value of the class. So the XGBoost uses a tree learning algorithm where, if you see here, the verbose value is set to 1 in the first two nodes to print the evaluation metric, which enables us to check the progress of the training process. And here, the verbose is actually set to 2, if you see. And so this is to print the tree information, details of which I have included in my final report. So I ran the model for both the test data set and the training data set. And once after I ran the model, so this is for the training data set, and then I repeat the same steps for my test data set. And finally, once after I finish running the model, I have so this is what the XGBoost model predicted for me. So for this, you can identify, and the training accuracy of the model was 31.78%, and the final results with which the model predicted were. So you see the ID 0. So for ID 0, the mutant, it belongs to the class 7, and ID 1 belongs to class 4. So it predicts which of these mutants or which of these variants belong to which class of mutants. So again, this is predicted using the XGBoost model, which we are going to compare it with the results of the Python Keras model, which I'll explain in a minute. So I have included my Python script as a Python Jupyter notebook for which you need to have Jupyter Notebook installed in your system. So just go to command line prompt, type Jupyter Notebook, and your kernel, so the localhost should, you should be able to load it into site. And this is my Python Jupyter Notebook for the Keras model. So again, I read the data files into the model. So sorry. And So here you can see the variant info, the training info, and so the test variant info, and then the length of the variant gene. And so I'm directly moving to the model, which is okay. So I import the Keras package, and so you have to previously have it installed. And so once before loading the data, we clean the data using the doctor vector, which converts the words in a sentence into a vector. So after feeding data to the doctor vector, we must train the model. So the model is usually better trained if in each training epoch the sentences or the sequence of sentences fed to the model is randomized. So the model is trained here for 5 epochs and the model is saved to avoid retraining when it's loaded again. I also create a NumPy array as the classifier. The sklearn.model takes only NumPy arrays. So moving to the neural network model Keras. So a little bit of background info about that. So Keras is a high-level neural networks API that's written in Python, and the core data structure of Keras is a model, a way to organize layers. So you can stack layer, and you can create additional layers. I chose the sequential model, which is a simple model with a linear stack of layers. And so here I define the model, and then once after you define the model, you compile the model, and then you fit the model. Yes. And so here we achieve the training accuracy of like 56.63% using the Keras model. And so this is the graph that we obtained from Matplotlib that shows the model accuracy and model loss. So moving on to the final results. So the training accuracy using the XGBoost model was 31.78%, whereas the training accuracy that I achieved using the Keras model was 56.63%, and the validation accuracy using Keras model was 35.94%. So one of the reasons why the training accuracy would be less as the data set is like relatively small. So if we have more clinical evidence, we could create a bigger yeah. So now we are comparing the results that we got from the predictions of the XGBoost model and the Python model. So if you see here for ID 0, R has predicted class 7, which you can see the same in Python, 0.65 that has the highest value, and ID 1 class 4, and here ID 1 has like 0.7. So the prediction results of almost both the models are same, except that the accuracy is different between both the models.